Okay, let's go ahead and get started. I'd like to first start off by re-explaining something that I may have misstated or may have been a bit confusing yesterday, and that has to do with the expression of the MHC class 1 molecule. And so I'll just restate how it should be. Basically what I said is you can express up to 6 different MHC class 1 molecules. And I may have stated that each cell can only express one type. That's actually not true. When we talk about expressing 6 different MHC class 1 molecules, we're talking about the HLA class, HLA A, B, and C. So these are the MHC class 1 molecules that can express the antigen. And as it turns out, as most of you know from basic genetics, that each cell has a set of genes from your mother as well as your father. And in those genes are genes coding for the different MHC class 1 molecules. So you can express one from your father as well as one from your mother. And there are 3 of them that express the antigens, A, B, and C. So you can express 1A from your father, 1A from your mother, 1B from your father, 1B from your mother, and so forth and so on. And it turns out you can express up to 6. You don't have to express 6 different ones because you can inherit the same MHC class 1 molecule from your mother and your father. So obviously if you inherit the same one, you want to express 6, you express less than 6. Does that make sense? So that's also what is meant by codominant. So both can be expressed at the same time. All right, so we can go ahead and start with today's lecture. We'll be discussing a number of different processes involved in the immune response. We'll start off by looking at the concept of antigen processing and presentation. So the question that arises, how do these peptides get into the MHC class 1 or class 2 group? How's that association, how does that happen? Talk a little more about co-stimulation and how that and why that's important for the development of the immune response. We'll look at a little bit of the self-MHC restriction. Then we'll spend a little time talking about the role of the thymus and how that is very important in the education of the T cells. And finally, we'll just spend a brief amount of time discussing superantigens and how the response to superantigens can differ from the response to conventional antigens. So the first thing we will look at is how does the ability of the B cell to recognize different peptides or antigens differ from the ability of the T cells. And most of this lies in the fact that the T cells have to recognize the antigen in association with MHC class 1 or class 2 molecules, whereas the B cells can recognize, they don't need this interaction, and they can recognize a number of different types of antigens. For example, they can recognize proteins, and here they can recognize a number of different proteins, different forms of proteins. It can be conformational determinant on that protein, so how it folds. They can look at denatured parts of the proteins, or even proteolyzed determinants. So they can recognize a number of different proteins and a number of different forms of the proteins. They can also recognize nucleic acids, polysaccharides, as well as some lipids. So the diversity of which the antibodies can recognize antigens is much greater than, say, what a T cell can recognize. And finally, they can recognize small chemicals and haptens. And we'll discuss this a little bit later as well. So what can a T cell receptor recognize? It turns out that T cell receptor is very limited in what it can recognize. It can only recognize fragments of proteins associated with the MHC class 1 molecule. And for the most part, they don't recognize lipids, DNA. Typically, the only thing they can recognize is fragments of proteins. And as we discussed before, the helper T cells recognize the peptide in association with MHC class 2 molecules, so helper T cells class 2 MHC, and the cytotoxic T cells recognize the peptide in association with a class 1 MHC. Now, turns out that this association between the MHC molecule and the peptide is not a random event. It's a highly ordered process that actually takes some time. And if you look at, and this was determined experimentally, so we'll have two sets of experiments here. And in the first set, you can pulse a macrophage with a protein antigen. You wait a very short time. You wash the cells, so you get rid of all the external antigen, and you fix the cell. So once you fix the cell, it's no longer able to do anything. Then you mix these cells, and you measure the proliferation. And it turns out, in this situation, you get no proliferation of the T cells. So the antigen is there, the antigen-presenting cell is there, everything is there, but you still don't get proliferation. Now, if you change this up a little bit, you pulse a macrophage with the same protein for a short time. You wash the cells. So you have some. When you wash the cells, you have some of the antigen that was taken up by the cells. You wait 4 to 5 hours. So the big difference here between these two situations is here you wait a very short time. Or you fix immediately, so it's a very short time. Here you fix after 4 to 5 hours. So you're giving these cells some time to actually do something with this antigen. Then you mix it with the T cells and you measure proliferation, and it turns out you get proliferation of the cells. But it turns out that these cells need some time to do something with this antigen in order to present it with the MHC class molecule. And we'll discuss a little bit later what these processes are. So there are key steps in the T cell activation, a number of key steps. We'll discuss the APC's ability to process and present the peptide to the T cell, after which the T cell will receive a co-stimulatory signal. In addition to this, we'll see that in addition to receiving the signal and the co-stimulation, that it's very important for there to be adhesion molecules involved to stabilize the interaction between a T cell and APC. Once this all happens, then the T cell will receive the signal. The signal will be transmitted to the nucleus, and then cytokines will be produced to help drive the cell division. And we'll also discuss some of the cytokines that are produced and their role in the immune response in the second lecture. Shown here again is just a cartoon of the key steps involved in T cell activation. So it's not just simply processing and presenting an antigen to the T cell. In addition to that, there are a number of other processes that must take place in order for the T cell to actually become activated. One of the first steps is the adhesion of the T cell with the APC. So something has to stop them, something has to allow them to bind to one another. After which, the T cell must recognize the peptide in association with the MHC class 1 or class 2 molecule. After these first two steps take place, there must be co-stimulation. So there has to be another signal that's transmitted from the APC to the T cell. But once you receive these three steps happen, only then will the T cell become activated and produce the cytokines. Sure. Typically, no. Yes. Throughout your body, the cells are constantly coming into contact with one another and checking out. Basically, the T cell will go and basically check out what's being expressed by the other cells. Again, this is the same cartoon, just shown a little bit differently. Adhesion, T cell receptor, ligation with the appropriate MHC class 1 molecule, co-stimulation, and then you get production of various cytokines, which leads to the stimulation of these cells. So before all this can happen, you have to have the peptide associated with the MHC class 1 or class 2 molecule. So how does that happen? And there's some basic steps that take place. First, there has to be fragmentation of the protein of the peptide. So your bacteria or your other pathogen floating around in your blood, it's a whole organism. It's not fragments. And the T cell, or the APCs are not able to present the whole bacteria. They actually can only present very small fragments. So these different pathogens, the proteins in these pathogens have to be fragmented. Then these fragments have to associate with MHC class 1 or class 2 molecules. Once that happens, then you need transport to the cell surface, because association with the MHC class 1 molecule is not enough. You actually have to have these MHC class 1 molecules present on the surface. Yes? Typically, the APCs will deal with extracellular bacteria, but in the case of a virus, some of the viral proteins that are produced will be degraded within the cell. But we'll go over that a little bit later. And again, there are different cellular pathways for the association of the class 1 and class 2. That's kind of alluding to your question. So the class 1 deals with peptides in a certain way, and the class 2 will deal with peptides in a different way. And we'll go over the differences in the next few slides. So here's the MHC class 1 molecule. And typically, for a peptide to be associated with a class 1 MHC molecule, the peptides have to be cytosolic. So given here is an example of a viral infection. Typically, when a virus infects a cell, it'll use part of that cell's machinery to produce different proteins. So shown here is the viral proteins being produced. Once these are produced and released into the cytosol, part of these proteins are actually taken up into a proteasome. And a proteasome is basically a compartment full of enzymes that are capable of cleaving proteins. They have a number of different proteases in these compartments. So they'll chew up the protein. Once these proteins are chewed up, they're released. And the next problem is, once they're released into the cytosol, where do they go? And it turns out that in order for these peptides to be associated with the MHC class 1 molecule, they have to get into the endoplasmic reticulum, because that's where the MHC class 1 molecules are produced. And it turns out there are a couple of proteins that act as peptide transporters. So you have release of these degraded proteins into the cytosol. They're then able to get into the endoplasmic reticulum through these peptide transporters. Once they're in the endoplasmic reticulum, they're able to associate with the MHC class 1 molecule. Once this process happens, the MHC class 1 molecule will be transported into the Golgi apparatus, and then finally it will make its way up to the cell surface. Once it gets to the cell surface, it is then able to be recognized by a T cell expressing the appropriate T cell receptor. An interesting point here, and I think we mentioned this before, that certain cytokines can lead to the increased expression of MHC class 1 molecules. So if you have an immune response, some cytokines can be produced that lead to increased regulation of MHC class 1 molecule. Now, in addition, a number of these proteasomes, or these proteases found in the proteasome, as well as these peptide transporters, they're also associated, the genes for the expression of these proteins is also associated with the genes for the expression of MHC class 1 molecule. So in many cases, if you get increased expression of MHC class 1 molecule, you'll get increased expression of the specific proteases, as well as increased expression of the peptide transporters. So the whole machinery gets ramped up. Is there a question?
Is there a that proteasome know what protein?
Typically, no. What happens is this is very, very nonspecific. There are some proteases that cleave certain length peptides, so they cleave 8 to 10 length peptides. But what happens here is even your self-peptides are cleaved and produced and presented in the same way. So this process here is very nonspecific. It doesn't know that you're infected with the virus. It doesn't know what's going on. It is typically presenting all the peptides that can be found in that cell. The specificity resides in the ability of the T cell to recognize it as foreign. Any other questions? Yes. What happens is there are other processes involved in immune response. So, for example, if another cell is activated, it may produce cytokines, and there are certain cytokines that will actually lead to the upregulation of MHC class 1 molecule. that clear? So it's kind of a loop effect. Okay. So the class 2 the generation or the processing and presentation by the MHC class 2 or the APCs is a little bit different. As we mentioned before, the APCs typically will sample what kind of proteins are in the extracellular environment, and they typically deal with extracellular bacteria and other pathogens like that. And so these proteins are actually not produced within the cell. They're actually extracellular. So there has to be a process for getting these proteins into the cell. Now what happens is that many of the APCs are able to endocytose these different proteins. So basically they'll gobble up the proteins. Once they bring them into the intracellular space in what is called an endosome, the endosome can interact with the lysosome, which creates a situation in which these peptides can be degraded. Now, when we look at the MHC class 2 molecule, it is also synthesized in the endoplasmic reticulum. And it's basically synthesized at the same time or the same space that the MHC class 1 molecule is produced. And in this case, you have, in a cell that has the MHC class 2 molecule being produced, you also have the MHC class 1 molecule being produced. Therefore, within this space, you also have the peptides that may want to bind, that may bind with the MHC class 1 molecule. The question is, how do you keep those peptides from binding to the MHC class 2 molecule? Again, because the only ones you want binding to the MHC class 2 molecule are the ones found up here in these endosomes. As it turns out that in addition to the alpha and beta chain of the MHC class 2 molecule, there's a third chain known as the invariant chain that is produced. And this particular chain serves two functions. The first of which is that when it binds to associates with the alpha and the beta, this chain kind of loops over top of the binding groove. So it blocks the binding groove. So no peptides can be associated with the MHC class 2 molecule in this situation. After which, again, this MHC class 2 molecule is produced. It will also travel to the Golgi apparatus. And the second function of the invariant chain is that instead of telling this MHC class 2 molecule to go directly to the cell surface, it actually gives it a signal and tells it to fuse with an endosome. So the endosome has all the different peptides found in the extracellular space. These two fuse. And as we mentioned, this environment here is very, I guess it's pretty lytic. And what happens is when these two fuse, this environment becomes such that this invariant chain is now cleaved off of the MHC class 2 molecule. You have a free MHC class 2 molecule, and in this same compartment you have the different peptides. These peptides are then able to associate with the MHC class 2 molecule, and they are then transported to the cell surface, where they can interact with a CD4 helper T cell. So if you want to compare and contrast the two processes, again, both MHC class 1, shown here, and the class 2 are synthesized in the endoplasmic reticulum. Found in this endoplasmic reticulum, there will be peptides, 8 to 10 amino acid length peptides, that then it can associate with the class 1 MHC. It cannot associate with MHC class 2 because of this invariant chain, which is blocking that binding groove. Both of these molecules then go to the Golgi apparatus, and this is where they start to differ even more. The class 1 molecule, which already has the peptide in its groove, goes straight up to the cell surface. The class 2 molecule then goes, because of the invariant chain, will then fuse with the endosome, and this endosome contains the peptides from the extracellular portion. In this fused vesicle, this is where you get cleavage to the invariant chain, and then the association with the new peptide. This MHC class 2 molecule with the peptide then goes to the cell surface, where it can interact with the T helper cell. So are those differences clear? Okay. Yes. Typically, yes.
We that in the last lecture.
Anything typically the class 2, anything that associates with the class 2 is typically from an extracellular infection.
Is it, and once it's become
That's a pretty complicated question, but typically the vesicular infection in most cases will involve the association with like a lysosome or an endosome. Yeah, I think that someone else had the same question, and I responded with an email. Hopefully it was a little more clear, but yeah. So again, this is again a cartoon of the MHC class, the association of the MHC class 2 molecule, and how it interacts with the peptide. So again, when it's first generated, it has this invariant chain. You see, shown here are the different peptides, and they're unable to actually bind to this because this binding groove is being blocked with the invariant chain. This then progresses, and the invariant chain gets clipped off. Once it's released, it is then able to associate with the peptide in the endosome. Okay, so this is kind of goes back to your question. There's a number of points that are important for processing and presentation if you look at the difference between the class 1 and class 2. And the major one is the location of the pathogen. So where is the infection happening? Where is this pathogen residing? The viruses in the cytosol typically will associate with MHC class 1, and you get a T-side CTL or T-side toxic response. Extracellular bacteria will bind with MHC class 2, and that will develop a Th2 response, an antibody formation. Intracellular bacteria still will end up in those lysosomes and will elicit or bind to the MHC class 2 and develop into a Th1 response. The difference between a Th1 and Th2 becomes a little more complicated. Th1 will elicit more of the CTL response or T-side toxic response, and the Th2 is primarily a humoral response, antibodies. The vacuoles?
Yeah. We were talking about last lecture,
Yeah. The ones that get into the different vesicles, those typically will be degraded and associated with the class 2 molecule. Okay. This is also another point. Peptides derived from both the self and non-self proteins can associate with MHC class 1 and class 2 molecules. And this is important because each peptide, each time you have an MHC class 1 molecule in your cell, it has some peptide in it. And typically in a normal cell, this peptides will be self. The T cell will interact with it and recognize it as self and will not develop an immune response. And if you don't have MHC class 1 molecules on your cell surface, another type of cell will come along. Typically it's an NK cell. That cell will recognize this cell as being something wrong with this cell and will develop an immune response. And we discussed this before. The chemical nature of the MHC group determines which peptides it can bind. It has a lot to do with the anchor sites. So each MHC molecule has in it an anchor site that requires a particular amino acid. And if you don't have your amino acid in the right spot, the peptide won't bind. And here's another important point. T cells only recognize foreign antigen with self-MHC. So when the T cells, we'll talk about this a little bit later, but when T cells develop, one of the first things that they have to in the development is their ability to recognize self-MHC. And if they don't recognize self-MHC, they're quickly deleted from the body. Question? Yes. No, they don't no. They don't recognize self-proteins. We'll go over this a little bit more. I guess that's a bit confusing. During T-cell development in the thymus, and we'll discuss this a little bit later, one of the first steps is that the T-cell has to get through, one of the first loops, is it has to be able to recognize self-MHC. If it recognizes self-MHC, it's allowed to progress. If it does not, it's deleted. The next step is they check for the ability to recognize self-antigen. If they can recognize self-antigen, they're also deleted. If they can recognize self-antigen, that may lead to an autoimmune disorder. Okay, so that may have been a bit confusing. So the cells in your periphery cannot recognize the self-peptides. Okay? Again, there's no value in someone a T-cell being able to recognize foreign MHC. Again, there's a restriction that the way that the peptides are presented is with class 1 MHC or class 2 MHC, and the T-cell receptor recognizes both the MHC class molecule as well as the peptide. So if you produce T-cells that don't recognize MHC class molecules, they will not be effective in the immune response. And again, there are hundreds of different MHC class 1 and class 2 molecules. So if you just produce T-cells randomly, you'd have most of your T-cells would be ineffective, and it'd be a waste of energy. And we'll discuss this in much greater detail, but self-MHC restriction occurs in the thymus. T-cells basically migrate to the thymus, and that's where they get their education on what to and what not to recognize. And shown here is an experiment in which MHC restriction is demonstrated. Again, here you have two different strains of mice, strain A or B. Let's say this is strain A, strain B. And what happens if you immunize this mouse with an antigen, wait a period of time, so you have to give these mice time to process and present this antigen. You take the T cells from this mouse and put it into a Petri plate. Again, you have mouse strain A or B. You take out the macrophage. Now, the macrophage in this case are acting as the antigen-presenting cells. You mix them with that very same antigen and then put them back into this Petri plate. Well, you notice if you do some mixing and matching between the two. So you take the T cells from strain A and mix them with the macrophage from strain A. So these two will have the same MHC class 2 molecules. If you look at proliferation of these cells, it happens. Now, if you take macrophage, the APC from strain A, and mix it with the T cell from strain B, you have incompatible MHC class 1 or class 2 molecules here. So you do not get proliferation. Third case, if you take the macrophage of APC from the strain B, mix it with a T cell from strain B, again, you get proliferation. And then finally, if you take the macrophage or the APC from strain B and mix it with a T cell from A, you do not get it. So you have the exact same antigen, but unless you have the exact same MHC class 2 molecule, you do not get proliferation. And this is the same for the MHC class 1 molecules. Shown here is a very similar experiment. In this case, you'll immunize the mice with a virus. 7 days later, you'll take out the spleen cells, get the cytotoxic T cells out of there. From another mouse, you can take the fibroblast out and infect these fibroblasts with this virus. So fibroblasts are easily infected with virus. So here you have a virally infected cells associating viral proteins with the MHC class 1 molecule. And here you have the CTL that should be specific for this MHC class 1 molecule with this peptide. If you do the same type of mixing experiment, if you mix the CTLs from strain A with the target from strain A, you get killing. If you mix them up, either here or shown down here, you take CTLs from one and the virus or the fibroblast from the other one, you don't get killing. Again, B and B equals killing. So this basically what this demonstrates is that you need, in addition to having the same virus or the same antigen, you have to have the same MHC class 1 molecule or CTLs won't work either. Now let's talk a little bit about the antigen-presenting cells. What cell types can actually present antigen with class 2 MHC? And typically there are three main cell types. They are the dendritic cells, the macrophage, and the B cells. And these cells play a major role in the activation of naive T cells. And once you have the interaction between the naive T cells with appropriate APC, you get a prime T cell. And this prime T cell then has less requirement for these APCs. So first we can talk about the dendritic cells. These cells are found primarily in the T cell area, which makes sense. If you have a APC that's job is to present antigen to a T cell, it makes sense for it to be found in association with the T cell. However, there are also other specialized cells known as Langerhans cells, which are found in your skin. And their main job is to sample the different antigens in your skin. Typically, dendritic cells will pick up antigen in peripheral tissues, so they'll look for sites of infection. Then they'll migrate back to the lymph nodes. And in the lymph nodes, that's where they'll actually encounter the T cells. Interestingly here, a very interesting aspect of DCs, they can actually present peptide in association with class 2 or class 1. They can actually lead to cross-presentation and lead to direct activation of CTLs. Second population of APCs are the macrophage. You've heard a lot about these. And they primarily act by gobbling up or ingesting different microbes. These are then digested in the phagolysosome, and then they present the fragments, cell surface, again, in associated with MHC class 2 molecule. And the third type is the B cell. And this acts a little bit differently. You don't have the non-discriminate engulfing of microbes. They are a little more specialized. They actually bind to the different antigens using their surface immunoglobulins. It's more specific. After that binds to the surface, they then internalize the antigen. They then will degrade it, as we discussed, using the class 2 MHC pathway. And it turns out that during an infection, if the antigen concentration is very low, the B cells become the best APCs because they're very specialized. They can pick up very small amounts of antigen. Whereas the macrophage and dendritic cells more or less gobble up everything. So this is a summary of the three different primary APC types. You have the mononuclear phagocytes, which are typically your macrophage. They can actually ingest the different pathogens in a number of different ways. They can have some non-specific receptors that allow them to take up the pathogen, or they can do it following opsonization. So the microbe may have been labeled with the antibody. The Fc receptors can bind to, the Fc portion of these antibodies bind to Fc receptors. They become ingested and then processed and presented with class 2 MHC. B cells, again, they act by sampling the antigen, and only very specific antigens can be bound by the B cells. So the immunoglobulin receptor has to recognize a specific receptor. Once that happens, you have the same process. The B cells take up the antigen, process, and present it to the T helper cell. And finally, there's a dendritic cell, and it kind of looks like an octopus because of all these arms. And these arms are used to basically gobble up as much antigen as possible. They then process and present the same way, and using class 2 pathway, and present to the T helper cell. Is that clear? The three main types of APCs. You have the macrophage, B cells, and dendritic cells. Okay, so we've gone through a few. The first step, which is processing and presentation to the T cells, and that provides a first signal. Now, in addition to that, there has to be co-stimulation. And it turns out that B7, expressed by the APC, plays a very important role in this co-stimulation. and it interacts primarily with CD28 on the T cells. And it turns out that if you have ligation of the T cell receptor in the MHC class 1 or class 2 molecule, in the absence of co-stimulation, these cells become anergic. That is, not only is there not a response, but they basically become incapable of responding to future challenges. And that's shown basically here. Here are the three situations that can happen. In the first one, you have the MHC class 1 or class 2 molecule in association with a peptide interacting with a T cell receptor, but no co-stimulation. These cells become anergic. So basically, they're put to sleep. These cells are no longer able to recognize and become activated in response to these antigens. In the second situation, you can have just co-stimulation. So there will be a B7 expressed by your APC, CD28 expressed by the T cell, but no T cell receptor MHC class 1 molecule interaction. Again, you have no effect. There's no signal whatsoever here. And then the third is you get T cell receptor recognizing the MHC class 1 molecule with a peptide, followed by co-stimulation. You get activation and production of cytokines. Shown here, we're not going to really discuss this, but it's generally believed that in addition to CD28, there's a second molecule known as CTLA4, and it's generally believed that this molecule here can actually act to downregulate. The second signal here can actually downregulate the immune response. Yeah, so inactivate the cells. A bit of controversy regarding that. But the point you can take here is that B7 combined with CD28 as well as CTLA4. And typically, these will result in a different type of response. So shown here, you see, back here, you see that on this cartoon that you have this interaction here between the APC and the T cell. And it turns out in recent times they've shown that the junction between the T cell and the APC is actually a highly ordered complex. And they term this as an immunological synapse. Within the synapse, there are two defined regions. There's the peripheral supramolecular activation cluster. And they call it the PSMAC. Saying all that is way too much. And there's also the central supramolecular activation cluster, or the CSMAC. And typically, the PSMAC contains most of the adhesion molecules, and the CSMAC contains most of the signaling molecules. Shown here is a cartoon of a typical immunological synapse. Again, on the peripheral area, shown here, you have adhesion molecules, so ICAM and LFA. So these act to keep the cells together. In the central portion here, you have the T cell receptor binding to the class 1, for example, here, with the peptide, and you have the co-stimulatory molecules. The PSMAC is the peripheral area, so here and here. Typically, that's mostly adhesion molecules. And the central SMAC is the portion in the middle that has the signaling molecules. Shown here is a confocal micrograph, or an EM, of one interaction. Shown here in the blue is a T cell. Shown here in the green is the APC. And if you look in the middle here, the red area there, that's the peripheral SMAC. So that's the location of all mostly adhesion molecules. And the middle portion here is the central SMAC, and that's where the signal is being transduced. So I guess we spent a lot of time talking about conventional antigens, the normal way that most antigens are processed and presented. In addition to that, there are also antigens known as superantigens. And these are produced by a number of different bacteria. A couple of them are produced by staphylococcal. These do not require processing and presentation. In most cases, the intact protein, the protein is not chewed up, It will bind to a region of the T cell receptor in the MHC class II molecule on the APC, and it will lead to direct activation. Now, this leads to the activation of a large number of cells because there are only a limited number of T cell receptors, types of T cell receptors, and there are, or at least the part of the chain that the superantigens bind to is usually a bit more conserved, and there's no requirement for recognizing the antigen. Like I'll show you in the cartoon a little bit more what I'm talking about. So shown here is a conventional process or presentation. You have the antigen association with the class 1 and class 2, the class 2 shown here. And it has to be recognized by the T cell receptor, the variable portion of the T cell receptor. However, if you look at the superantigen, there's no requirement there. It basically binds to a portion of the beta 1 and the MHC class II molecule, and it binds to a portion of the T cell receptor. So there's no requirement here. So this region right here, as we discussed before, is not nearly as variable as this portion here. So these are more conserved than this region here. So you get activation of a large number of T cells. The exact purpose of this or the effect of this is not completely well known. In some cases, it's thought early on that by binding these, you actually end up deleting a lot of these particular cells. So typically activation of T cell eventually leads to the death of the T cell. So if you can bind to this portion and this portion of a large number of T cells, you can end up deleting those T cells. If you delete those specific T cells, they're no longer able to recognize and kill the bacteria. So it's kind of a defense mechanism for the bacteria. Medically speaking, these superantigens can cause a lot of problems because upon activating such a large number of T cells, you get a very high immune response. And that high immune response can lead to a number of disorders, can lead to some autoimmune-like disorders. Toxic shock is one. Acute respiratory distress syndrome is another. Running short on time. Last 5 minutes, I'll go through scientific education as much as possible. So the other part, you have the processing and presentation of the antigen. This is a non-specific process. The APC really doesn't know what antigen it's presenting. There's some requirements, but there's no real specificity there. The specificity in the immune response lies in the T cell receptor and the ability of the T cell to recognize the antigen. And the thymic, where this process takes place is typically in the thymus. And as discussed earlier, T cells, with T cell receptors recognizing self-MHC molecules, are retained. So the first requirement is that T cell must be able to recognize the MHC class 1 or class 2 molecules. These cells then go on to interact with other cells. And it turns out any T cell receptor that can recognize self-peptide in association with self-MHC, they're deleted. So these are the cells, if they're able to get out, these are the cells that will be able to recognize self-tissue, and these will can lead to autoimmune disorders. So these are cells you don't want circulating in your body. So then the T cells that can recognize self-MHC but don't recognize self-antigens are then allowed to circulate. It turns out that over 99% of the cells that enter the thymus do not leave because most of them are deleted. This is shown in a couple different ways. I'll skip over this slide and go to this slide. This basically describes the process of T cell maturation. When they enter the thymus, the T cells, the precursors are typically CD4, CD8 negative. So again, CD4 is a molecule that is primarily found on T helper cells, and the other population is CD8 cells, which are the T cytotoxic cells. When they first start out, they're neither. They go on to the next step. They will express some CD4 and some CD8, so they become double positive cells. The next step, they start to express some T cell receptor. This is when they start undergoing a positive and negative selection. Positive selection is any cell that can recognize self-MHC is positively selected. That's shown here. It can either recognize MHC class 1 or MHC class 2. If it does not recognize the MHC molecules, it's deleted by apoptosis. So already you've gotten rid of a lot of the cells. The next point is they then go on to interact with cells that are expressing self-peptides. So if the cell that can recognize MHC class 1 molecule, which will be a CD8 cell, then interacts with these cells expressing the self-peptide, and it recognizes the self-peptide, it gets deleted by apoptosis. Only the cells that can recognize MHC class 1 molecule and that don't recognize self-peptide, these cells are then allowed to circulate. When you look at the CD4 cell population, again, these have to recognize the MHC class 2 molecule. However, they cannot recognize any self-peptide in association with class 2. If they do, again, they'll delete it. If they don't, they're released into the mature T cell pool. So the majority of all cells that start this process are deleted because of one reason or another. Either they don't recognize MHC molecules, or they do recognize self-peptide. Yes, sorry. This right here is just part of the process. Once it's figured out if they recognize MHC class 1 or class 2, the next step is they become T cell receptor high. Because high and low means low expression of T cell receptor. High means higher expression of T cell receptor. And that's it. Are there any other questions?